Welcome to my presentation. My name is Natalia Ciesielska. I will be sharing my research on QTOF LCMS, a novel technique for the identification of decomposition chemicals of interest in aquatic environments. I hope you enjoy. Each year, human remains are discovered in aquatic environments due to accidents, natural and possible crime events. Worldwide drowning is estimated to be the third largest cause of injury and death globally. The United Kingdom in 2019 had 334 water-related deaths, following a concerning increase in water-related deaths last year of 631. Countless people become victims to water. Therefore, it is important to comprehend aquatic decomposition, which is still rather unknown. Criminal investigations and decomposition have a significant connection, as decomposition process can reveal valuable information that can aid an inquiry, such as help with searching for unrecorded burials, estimating time since death, PMI, cause and place of death. Estimating time since death is relevant to help identify human remains, contribute to cause of death, evaluate alibis of suspects, check witness accounts, and if remains are unidentified, they may help confine missing persons records. Also, compounds released by decomposing remains can contribute to manufacture a device which can detect cadavers or in the training of human remains detection dogs, supporting the search of a body in a criminal investigation. Animal analogues are frequently used as human proxies in forensic decomposition studies because of ethical restrictions surrounding the UK Human Tissue Act. A variety of substitutes are used for taphonomic research, including pigs, cows, sheep, rabbits, rats, and many others. Not all conditions and types of lifestyles are possible to study with animal analogues. Therefore, to close the knowledge gap, the UK would benefit from a human taphonomy facility. This project predominantly aimed to explore the use of QTOF LCMS for identification of decomposition chemicals of interest in aquatic environments, producing results in computer software qual. The first aim was to carry out untargeted water analysis using QTOF LCMS. The second aim was to track the presence of chemicals of interest found during decomposition in an aqueous environment over time. The hypothesis attempts to show if there are any chemicals of interest released in murine decomposition captured through application of untargeted approach. Method consisted of submerging 5 mice weighing approximately 17 to 25 grams in plastic containers with 400 ml of deionized water, including 2 negative controls, keeping lids on to minimize evaporation and contamination. A constant temperature of 20 degrees was maintained by an incubator. Over 8 weeks, 1 ml of water from the middle of each container was taken and filtered straight into vials. At the same time as sampling, pH and conductivity readings were measured. During the first 2 weeks, samples were collected daily. Following slowing down of erratic decomposition changes, sampling was decreased to interchanging days for another 2 weeks. Then, twice a week for the final 2 weeks. Photographs were taken of mice after sampling and TADS method applied to measure the decomposition process. This model divides the body into 3 sections as not all areas demonstrate the same aspects of decomposition. All sections have scores assigned to stages. Lastly, water samples were analysed using QTOF LCMS. Results can be divided into QTOF LCMS, pH, conductivity, and TADS. The compounds released after death are products of decomposition. Over 400 compounds in total were identified in the experiment with QTOF LCMS. Only 31 chemicals were of interest presented on the right-hand side. These were byproducts of mice decomposition, which only appeared in mice samples and not in any of the controls. Various chemical classes were found. 11 human metabolites, 8 amino acids, 2 enzymes, 3 proteins, and 1 rat metabolite and 6 others. Water pH at the start was acidic, 4.28, gradually increasing to be more alkaline, 7.0. Graph shows decomposition progress was quickly progressing from day 3 to 16, showing increase in pH, indicating samples became more alkaline. After 16 days, most pH remained moderately stable. The graph on the right-hand side shows water electrode conductivity change over time, displaying a comparison among all 7 samples. From day 0 through to 16, all 5 mice had a strong relationship based on gradually increasing values. A similar trend is reflected by pH results, also progressively increasing between days 3 and 16. But after day 16, a significant difference can be observed among each mouse, starting to display varying conductivity results. This is related to breakdown of molecules within the mouse, resulting in an increase of electrode conductivity, detecting electrolytes and ions from decomposition process. Overall, TAD scores increase from start to finish, but the most drastic visual changes occur between days 0 to 6 post-decomposition. After 6 days, decomposition slowed down, and little change was observed until day 14, displaying a rise again till day 20. After day 20, scores stayed constant, and mice presented very little decomposition change. However, only mouse 2 on day 26 and mouse 3 on day 30 went up. In conclusion, this experiment successfully demonstrated that QTOF LCMS used for untargeted analysis to identify chemicals of interest released by mouse decomposition in aqueous environments is a powerful detection technique. The untargeted searches identified complex chemical mixtures, identifying 31 chemicals of interest. These multiple chemicals established provide important information and better understanding of murine decomposition, facilitating new information of mammal analogues in taphonomy. There is a clear difference in compounds released during decomposition of mice carcasses, humans, and other animal species. The results show that only 1 chemical of interest, decanol, from this experiment was also identified by Vass 2008 in a previous human decomposition study. Despite decanol being a human metabolite, it is not a human-specific marker, since it's commonly identified in dog, pig, and deer decomposition. However, stearic acid was also observed in mice aqueous decomposition, found to be a mouse metabolite, which was identified as a product of body decaying during decomposition, but no other decomposition research identified stearic acid as a human or animal decomposition marker. Consequently, these results may indicate potential new biomarkers for the identification of specific post-mortem circumstances that can offer new knowledge and better understanding into investigations related to missing persons in aquatic environments. To date, there is little investigation into murine aquatic decomposition. Future work should consider testing the QTOF LCMS technique for identification of human and other animal species decomposition chemicals of interest in aquatic environments. Because with more research, it may be possible to use animal models as correct substitutes for human bodies to evaluate certain areas of decomposition. With further research, it could be achievable to adapt experiments using animal models to simulate certain aspects of human decomposition more accurately. Also, experiments should be conducted in less artificial experiments to simulate a more realistic environment. Thank you for listening to my presentation. I hope you all enjoyed it.